So when we work in our lab, we usually have, all our supplies on our workstation that we work with. We always want to wear safety glasses when we are working with anything in our lab. That's requirement from the safety office. So we get to look cool and sciencey. So we are going to be working with that. Now I have over here some tubes that we normally use in our lab this week or when we do the pipetting lab to make some dilutions. We usually just have some glass tubes that we work with. We wash them, the students wash them at the end of the day so they can be used by another class. I have some water and I have some dye, colored water so we can make dilutions. I also went ahead and brought some centrifuge tubes over for you guys to look at. So these are super small centrifuge tubes, right? So we can have them in all different kinds of sizes. Many times when we are working in molecular and cellular biology lab, we are using small tubes. Usually they are either these 0.6 ml tubes or these 1.6 ml tubes. So you can see the difference in size between those two. Other times we use 2 ml tubes, which are slightly larger. And then when we do DNA extraction, we use these big centrifuge tubes because we have larger sample size to work with to begin with initially. And these are 5 ml centrifuge tubes. So these are some of the tubes that we work with. Usually when we are making dilutions to prepare standard curves or anytime, like we have samples that we are diluting, we have super tiny amounts. So we typically only use these little tiny tubes to make our dilutions in. So today I'm going to do 2 sets of dilutions for you to look at. And in doing that, I'll be demonstrating the actual technique. One is going to be with these serological pipettes. So these are serological pipettes. The one I have in my hand is a glass pipette. So these can be washed and reused. Other times when we're doing aseptic technique, we use serological pipettes that are individually wrapped. They're usually plastic. They're one-time use or a couple times while we are working with them right there in aseptic space, and then we throw them away in biohazard waste at the end of the day, since they usually have cell components in them. So these are the ones that I just brought in because we're just using some color water and dye, and that's it. And these are basically like little mini cylinders, right? So if you look at them, they have gradations and numbers on one side. So if you look at the numbers, they go from, I don't know if you can see the numbers, unfortunately. There. Oh, now I know why they do that in all those YouTube videos. So there you go. So if you look at them like this, right, the numbers are going from top being 0 all the way to the bottom being 10 ml. And if I look at the numbers on the opposite side, oh wow, I learned a new technique from a YouTuber. Oh nice. They go the other way, right? So these numbers, if you were to look at them closely, they actually go the right way. So they start off on 10. And so from the bottom, they go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now, I wish they made the bold side the one that goes the correct way, because it's easier for people to usually just measure out what they want to measure out instead of having to calculate it out from this side. So if you wanted to pick up 3 ml, you would lift it up to 7. But if you were going the other side, you would just pick it up to 3 like a normal person. But that's just a little minor issue with these. Now, these serological pipettes, they are meant to be used so that you dispense everything that's in the actual tip as well. But the last, these, half an ml that goes in this last little bit is not graded. So you can accurately pipette up to 9.5 ml, and then that half ml, if you wanted to dispense 9.7, you would be just guesstimating. So that's just something to be aware of. That these are pretty accurate, but only up to that last half ml. Okay, so these are what we're gonna use. So this is like a mini graduated cylinder, like I said, so you would read it just like you would read a graduated cylinder at the bottom of the meniscus. You use these pumps with them. They come in different sizes and different colors to identify what they are for. These green ones can be used all the way up to 10 ml. We have blue ones that go on the smaller, tinier pipettes. They go up to 1 to 2 ml. And then we have larger, this kind of my shirt color ones that go all the way up to 25 ml. But most of the time we are using these green ones because they're quite versatile, and usually the liquids that we are working with are no more than 10 mLs. Now, since I only have one of these to play with, I'm going to be doing my water first, and then I would go ahead and add my dye afterwards. So when you're playing with them, or working with them, rather, you are going to be taking in your liquid from the tube. And usually what I do is I keep one pipette that's strictly for my colored solution or for my chemical, and one that is strictly for my diluent or water. In this case, since I'm doing the water first, I'm just gonna be, I'm using one, so I'm just doing water first. So now I'm gonna make a 1/2 dilution series. So that means that I'm going to be diluting each sample before by half. So if I start off with 100% blue solution, the second one is going to be 50% blue, then 25% blue, then 12.5% blue, and then 6.25% blue. Okay, so I'm going to leave my first tube alone since that's going to be 100% blue solution. My son is writing me a note from across the room that he made the dyes for me. I did not, and that is true. If I had made them, they would have been like red and blue, so I could make purple. But he made blue and black. So I know I never told you that. I thought to see your own creativity. Okay, so I'm gonna go ahead and take 2.5 ml in here. And again, you want to read it at the bottom of the meniscus. When you have picked it up appropriately, you're going to notice there'll be no leakage from the tip. There should be nothing dripping off. And I'll show you in a second how it is that you're actually be doing that. And then I will release it where I want. Now, when I release, you notice that there is some stuff left over in the tip. Well, we need to get rid of that stuff. And what we do is we go pump it up and down, which I will show you in the next step, to release all the liquid. When we pump it up and down really quickly, you notice that there is a lot less liquid left behind. Now, I will show you how it is that we did that. So I'm going to move it up a little bit. I'm actually in my son's room today, so that's why you see some of the little kiddo paraphernalia over there. Way to turn it to make it less paraphernalia, but that's okay. Okay, so this is your pump. This has many little dials and stuff. So you can dial it up. When I dial it down, it goes up. That means the liquid's going to go in. When I move it up, dial it up, the liquid is going to go down because the plunger is moving down. You also have this little button that you can use to dispense the liquid, or you can actually press it down from this top plunger. You can, press the actual plunger down as well to dispense the liquid. So there are multiple ways that you can dispense liquid. The only way to pick up the liquid, however, is to dial it up. Now, to fit the pipettes, because different companies' pipettes are slightly different in dimensions, when we insert them, we want to firmly insert them. And then we want to tighten this a little bit if needed, or loosen it if needed, to make sure it's a snug fit, but not, not so loose that it drops down and not so tight that it breaks. So we do that to fit it. Now, when we're working with them, we want to work with them vertically at all times, or most, we don't want to tilt it too much because that's going to destroy our accuracy. To keep it accurate, you always want to work vertically as much as possible. Secondly, when these are in your hand, you don't want to be going like this and talking to each other because, you're essentially, that's not safe. You're going to stab somebody in their face, in their eyes, and then they will complain, and that will not be a good choice. Okay, so I'm going to now show you again, and I guess I have to move it further up, don't I? Yes. So I'm going to pick up my 2.5 by dialing it down So it lifts up my liquid. I get the amount that I need. I look at it, it's not dripping. I go to my second tube now, and I am going to release it using the side button this time, so I can show you the difference. So when I do that, you can look at it and you see there's actually more liquid left behind, and that's true. The plunger is not as good at getting all the liquid out as you need. So let's fix that. To fix it, you basically go up and down super quickly with the side dial. And now, just a few times, and you can notice there is less liquid left behind. Okay. Okay, so I'm gonna do one more just to show you, and then I will let it be. So there, my 2.5. And this time, maybe I'll use the top plunger. And so you will actually notice that's the best way to get majority of it out the first time. But still, I'm gonna make sure and double check and get everything else out. Okay, in between steps, it's a good habit to like take it out and push it back in so there's no air bubble left behind that may disturb your dilution series when you are mixing them up. Any questions so far? Can you guys see it, by the way, well enough? Yes, no, maybe. You can just send me a little thing in the chat. Sounds good. Okay, so of course, this is something that you may have all worked with already, which is great, but you know, this is a review plus giving you some extra pointers to get your accuracy correct. In Mosel, it's very, very important to be extremely accurate. Since a lot of our assays depend on how well we do the work technically in order to get a good quantification or in order to get good data from that experiment. Since usually we're working with super tiny volumes anyway. Okay, so now, because I was doing that, I'm going to now go ahead and take my colored solution for the first one. So you guys can go ahead. I was going to make a poll. I forgot how to do it. I will have to relearn. So instead, I would like you guys to just write the answer in the chat. We added 2.5 mLs of liquid of water in our dilution series. So how much of the dye should I take in the very first tube in order to be, so that I can make a 50% dilution for the rest of them? Very first tube, how much of the dye should I get in there? And I will wait for your answers before I actually pick it up. If I take 2.5 ml, then I'll have nothing left for my first tube, would I? So think about it a little bit. So how much should I get instead? So the question is, I want to make a 50% dilution series with the blue dye. I already added the diluent in my 3 tubes, and the diluent that I added was 2.5 ml per tube. Now I want to take my blue dye and dilute it by 50%. So in the very first tube, I'm going to have 100% blue dye. How much of the 100% blue dye should I put in here so that I can make a 2.5, a 50% dilution series from this tube into the other tubes? You guys are on the right track, but in the opposite direction. Remember, I need 2.5 ml. Since I need to have equal volume in all tubes at the end, I need to have 2.5 ml at minimum in that first tube. Yes, that's it, 5 ml. Because the first 2.5 is just what I need to get the numbers from my 100%. I'll need another 2.5. There you go, you got it. so that I can make the total. So since I already put 2.5 in there, I'm going to go ahead and put another 2.5 in there. And now I'm ready to make my dilution series. Okay, so once you have 5 ml in the first of the 100% dye and 2.5 ml of water in the rest, you're ready for your dilution series. I'm going to take 2.5 ml from my first tube, which is my 100% blue dye. And I'm going to put it in my second tube. And then I mix it. So I mix it simply by pipetting everything up in there and then dispensing it back down. And that makes sure that I get it well mixed and get a homogeneous solution in there. This is especially important if you have, liquids with different viscosities. So think about it as, something like honey mixing in with water. If you just put it in, it's not going to mix. But if you pipette it up and down, it will mix and make a homogeneous solution. So you do that, then you release it, make sure this is empty, push it back in, and then because I'm making a serial dilution, I'm going to get 2.5 ml from my second tube and move it to my third. And now I repeat the process. I pick all the liquid up, I mix it a couple of times, and then I make sure all the liquid is out by pipetting up and down. Okay. And then release it, push it back. And now I'm going to repeat it one more time by taking 2.5 ml and putting it into my fourth tube. There we go. And then you mix it again by taking all the liquid and releasing it. Cool. So now I'm going to go ahead and put this down for a second. How can I tell that I have made my dilution series correctly? Let's first start with that. Oh, I was going to bring an open serve thing. Okay, I will. That's okay. I'll show you in a second. How can I tell qualitatively, so not quantitatively, qualitatively, that I have made the correct dilution series? All my dilutions were made accurately. How could I tell that? Again, you can use the chat. Yes, each tube except for the very first tube had 2.5 ml of water in it. Yes, that's correct. If I made them correctly, I can look at them and each time it should be less and less dilute, right? So I will see if I can hold for. That's one way that you can tell that you've made them correctly. The color should be diluting out. Now, there's another caveat to that, and this would have been better, obviously, if I had remembered to bring my open rack instead of the closed rack. You don't have it, baby. The other thing is that all the meniscuses, and I'm not holding them quite at the same level, so you won't be able to see, all the meniscuses should line up, except for the very last tube, which will have twice the amount, because we didn't take that last 2.5 ml out from there. So qualitatively, you can look at your tubes and you can make sure that you made them correctly because the color faded over time, over the dilutions, each time by half. And then you can also see that the meniscuses are lining up. Now that's all good, but that may or may not be, directly related to the actual accuracy of your numbers. How can I accurately, quantitatively prove that I made these correctly, that these are done well? Again, you can use the chat area to write your answers, or you can use the mic and answer. So if you want to quantify how well you made your dilutions, what could you do? How would I do that? How could I measure the concentration of each individual tube? Yes, that's exactly what you would do. You would want to, somehow measure the concentration and then use it to graph it. But how would I do that? I can't really weigh them because it's not like a sugar molecule in there, right? And you would need a very different way to do that for each particular molecule that you use. If you use sugar versus salt versus, something light like what I have is a blue dye. It's in, very, very tiny difference in weight. What would I do instead that can be essentially a standard way to do it? Yes, there is a machine in the lab. You're right. It is called spectroscopy. That is right. So we are going to use a spectrophotometer to look at the absorption of light. The way that works is that if you just have a clear tube, right? You just have an empty tube. The light's going to go through. There's nothing to stop it from or, bounce it around because it just, other than the glass outer and the glass actually of the tube. It will go through mostly on a straight path. Most of the light will get through and you will have almost zero absorbance. You'll have a very tiny difference in the number. Now, if you have just water in there, you'll have some water molecules. The light may hit it and get bounced around a little, but overall, it will still mostly go through. It's a clear liquid, you won't have a problem. Now, when you start adding living cells, even if they're clear, obviously, if you add cells or blue molecules or sugar molecules or salt molecules, whatever it is. Now there are things in there that when the light is passing through, it's going to start bouncing off of, right? And because it's bouncing off of them, a percentage of it is going to get what we call absorbed into our sample, and only a small percentage will get through. The more blue molecules are there, or the more cells are there, or the more sugar molecules are there, the more light will get absorbed and the less will get through. So if we were to look at a range of dilutions where we know the amount of blue molecules in each one or the amount of substance in each one, we could graph it by looking at concentration of our solution and putting the absorbance we get on the y-axis. Now the C1V1 is great to actually calculate the concentrations, but it's not going to help you as far as the absorbance is concerned. You can predict what you should get, but you don't know if that's what's there. So yes, the C1V1 equation is what we use to calculate how much blue and how much water we are putting in to make the 25% or 6.25%. But to look at the accuracy of that final dilution, you need to then go back and do a spectrophotometric analysis. So what you will get today in your data set is that I will be giving you a couple of data sets from, a similar dilution series, and you will be plotting it to look at how accurately those were prepared, which one was done better or worse between the two of them. Okay, so that's what we do with the spectrophotometric analysis, is we look at the absorbance and then we relate it back to our concentration. So in an ideal world, if my first one, let's say the 100% blue gives an absorbance of 1, then what should be the absorbance in my 50% sample? Again, you can use that. Perfect, 0.5. So if in a perfect world, you would get a linear relationship. You would get, in 100%, if it is 1, in 50%, it will be 0.5. In a 25%, it will be 0.25. So easy as far as that is concerned. Good job. Now, another thing, this is just a tip for later. When we work in our labs, in our actual labs, and we are placing all these things down after our experiments, you always want to make sure to keep the tips of your pipettes away from you. Again, this is to ensure that you don't get any contaminant from that onto you, and also to ensure that you don't get stabbed by anything that can harm you. So that is just an aside for this serological pipette. Now, when we get to the actual graph, I will talk a little bit more about the linear portion of the graph and parts that may not be part of it. But for now, this gives you the basic idea behind what we are going to be doing in our assignment for the week. We will be taking data from a series of serial dilution, and we will be plotting it for the concentration against the absorbance. And then looking at the trend function to see if it was done correctly. If you've done it correctly, you should see or if the data is correct or made appropriately, you should get a linear trend function that is nice and clean. Okay, so now we move on to our micropipettes. How many of you have used micropipettes before and are comfortable with them? So only respond yes if you're comfortable with them. Great. So, you know, at least a few people have it, or a third of the class has some experience with it. That's good. If we were in our lab, you know, we would have had a lot of experience with these. So everyone in this class would have gone out knowing these very well. Unfortunately, we won't get that practical experience, but I'm hoping with the simulations and all the other stuff we will do, at least you'll get the theoretical portion of it, and you will get a good idea on how they work based off of that information. Okay, so I brought two pipettes with me. I brought one that is yellow and one that is blue. So for each pipette, we typically have 4 in our lab on each station. They go from the smallest range to the highest range. Whenever you look at a pipette up close, you know, along the side, it gives you the range that it can be used for. For example, the yellow pipette right here, it says that it can be used for 20 to 200 microliters. The blue one that's in my hand right here can be used for, I couldn't see, well, I know what it is, but I can somehow seem to get it to focus, but it's 100 to 1,000 microliters. So if it can go up to 1,000 microliter, how many ml is that? Good job. Yes, it is 1 ml. Okay, so to change those numbers, so this portion is essentially the pump portion. If you think about it from the serological pipette, this whole thing is the pump. And then to lift the liquid, we have tips that go on top of them. These tips come in boxes. They are plastic tips. They are usually clean, but they are not RNAs, DNAs free. They're not sterile. You can get sterile tips as well. If they're not, we can autoclave them, right? And when we autoclave them, we put a little tape indicator to, you know, tape the lid down, which is clear before autoclaving. And once the autoclave has cleaned them and sterilized them, they turn into these brown bars on top of them. So we know that the tips inside are sterile. But they're only sterile as long as you keep them there, right? As soon as I leave my lid open and just kind of hang out, then they're no longer sterile. But if I'm being careful and just taking out one at a time and closing the lid each time, then they remain clean. For many of our experiments, we don't really need sterile tips. We just need clean tips. So we are easily able to work with them and then, you know, refill up our boxes like we do in our class all the time. But for aseptic technique, when we are adding drugs to our cells or doing other things related to, you know, our cell culture, we need actual sterile, clean tips that are DNAs, RNAs free, that are without contamination of any sort, that won't have bacteria or hand cells on them, so they can keep our cultures clean. In that case, we provide you with special autoclave tips. Okay, so the tips here that I have 2 different varieties with my 2 pipettes. I have the yellow tips and then I have blue tips. The blue tips, obviously. So this is actually the cylinder portion of our thing, right? So this one can pick up to, I say 200 microliters, I believe. And this one can pick up to 1 ml. So these are the tips that we would use when we are working with our cell culture, with our micropipettes. Now, to change the numbers, there is a dial on each one of our micropipettes that tells where we are currently, and then we can change the numbers by dialing it back or forward. You never want to go below or above the limits of your pipette because it destroys the calibration and then it's no good, right? Then you might as well be just pipetting by hand. But this is how you change them. You can go backwards away from you and the numbers go down. You go move it towards you and the numbers go up. So I will show you, I'll go to the lowest number that this pipette can do. Maybe that will be better with the big one. I'll do the big one. I think it will show it better. So, you know, when you're moving them, you don't want to go like, that's not a good idea. Sometimes people do that and then they accidentally go over, or they are so rough with it that they break, and they are hard to fix. And they're expensive, so we don't want to do that. We want to be a little bit gentle with them. So I'm going to go to the lowest number it can get to, which is 100. Okay, so now I've set it to 100. Now on top, there is this dial that you can press. Right now you see that there is, you know, a little bit showing right now because I've only set it to 100 microliters. And when I press it, it will go down, right? But you still see a little bit. And then I press it again and it goes all the way down. Now, if you think about it in terms of serological pipettes, that first stop is the calibrated stop. So that dictates how much liquid I'm going to be picking up. When I'm releasing the liquid, if I just go down to that first stop, I won't get everything out. There'll still be a little liquid left behind. To do the same thing as pipetting up and down quickly to release all the liquid, I have to press all the way down. Okay, so I will show you what it would look like using these. Now these pipettes tips don't have any gradations on them. To pick up the pipette tip, you simply just like put it inside a pipette tip and press a little bit and they go on top. When you take it off, it shouldn't fall off. Simple as that. To pick up the liquid, you press to that. I'm trying to see. Well, maybe I'll do it with my left hand so you can see a little bit better. So you're going to press to the first stop, okay, before you put it into the liquid. You then go into the liquid that you want to pick up and you release it slowly up. Now, you don't want to go all the way down in the liquid. There are reasons for that, okay? When you take it out first, right, you see that there's a little bit of liquid in there, and that's all that I need because I'm only picking up 100 microliters. Now, with this, it shouldn't drip, right? If it's dripping, that knows it wasn't put properly and you need to do it again. There also should not be any liquid on the outside of the tip. You're working with such small volumes that you could easily have more than the volume inside on the outside of the tip. That's why you don't push it all the way into the liquid. Again, this is especially important if you're working with things like honey or, you know, viscous solutions. So DNA and proteins can be very viscous. So that consistency like honey, because that can easily stick to the outside and you will lose your accuracy. So I'm going to go ahead and put it into one of my little microcentrifuge tube. So to do it, I'm going to press to the first stop and I see there's a little bit of liquid there. I press all the way down and you can see there's a little bit of liquid in there, and this is my 100 microliters. I'm going to close it and put it down. Now, again, with these, just like we did with the other pipettes, we want to keep them vertical as much as possible. When we are releasing liquid, again, to make sure that we don't get any liquid from the outside of the tip into the system, we go a little bit, tilt it just a little bit, and go to the edge of our pipette, our microcentrifuge tube, and release it. That way you minimize any contamination from the outside of the tip. Okay, now I'm going to take the same pipette and I'm going to change the numbers so that they are the highest this one can pick up. And that's going to be 1,000 microliters or 1 ml. So now I've set it to 1,000 microliters. You can see Now, typically, we want to get rid of this tip before we use the next one, right? So to release the tip, there is this lever on the side. We press it and it comes off. So we are not interacting with that tip in any way at any time, especially when working with biohazardous materials. So I'm going to take a brand new tip. Again, the same way. I'm going to press. Okay, so now look at this. Because I set it to 1,000, you see the difference? It's lifted up. The tips are made of special plastic that can be autoclaved. So they're essentially just polypropylene or plastic tips. So here, if you look at this, you see the difference. This time, this is lifted a lot more. So because this is the calibrated, this just dictates the calibrated stops. And so I'm picking up a lot more liquid. I've set it to 1,000 microliters. There's a lot of difference. So to get to that first stop, I'm going to have to go a lot, right? So I can pick up a lot more with it. So, you know, this is an aside, but as scientists, we talk about how much we care about the environment, and we want to keep it clean, and we want to reduce waste and stuff. But as scientists, we make a lot of waste because of the materials we work with, but also because of our, you know, negligence as well. So one of the things that we work with a lot when we are in actual lab is that I want people to be aware of their steps in the work, so that they can minimize the amount of waste that they produce. For example, if I wanted to, you know, take these dilutions and I wanted to put them in a plate, if I start at the highest dilution, I have to change my tip every single time. But I could easily reduce my waste by 3 tips simply by starting off with the most diluted sample. And then I could use the same tip to collect sample from all 4 of them at the same time, you know, one by one. So this is something that we do talk a lot about inside the lab, obviously. And I'll try to make sure to, you know, show you guys where we can reduce waste as well. So I picked up my 1 ml, and now you can see this is a lot more liquid. And I'm going to put it in here. Press all the way down. So press to the first stop and then press all the way down. And you can see the difference in there. So again, you release it by pressing on the side, it releases my tip. Okay, so in the lab, we actually have racks that these go on. So in between, we always want to put them on the rack like this. I hang it in front of you. Okay, so here are my liquids. So this one is 100 microliters. This one is 1,000 microliters. And you can clearly see the difference between them.